Okay, so last lecture we discussed some of the basic components of the nervous system. So the cells that make up the the nervous system, the components of the neuron itself. But we still need to classify neurons based on their structure, and we already did the function. So the autonomic versus the somatic, the motor versus the sensory, and then of course we have the central versus the peripheral nervous system. So make sure that you're understanding how to classify the nervous system and the neurons into those categories. But we still have to be able to classify the structural components. So once again, structure will dictate function, and we're going to have these three classifications of the neuron that will be found in different areas of the body. So the three different types are going to be the three you see here. There's a multipolar neuron, a bipolar neuron, and a unipolar neuron. So as you might be able to point out, if you just took the polar out, you have uni, which means one, and it's also known as pseudo-unipolar, which means that it is a fake one, and we'll get to that. Bipolar, so two poles. Multipolar is several poles. And what they're talking about is how many axons or how many arms go off of the cell body. And the cell body in the multipolar neuron, which is going to be your typical neuron, is going to have several dendrites coming off of the cell body, and then one axon that comes off. So you can see all of the different dendrites coming off of the cell body, like the snakes of Medusa's head. So each dendrite then will branch further so that it can interact with as many neurons and or receptor fields as possible. So if this is a neuron in the central nervous system, for example, all of these are going to interact with other neurons in that field. If this is a receptive neuron, then that would be the different dendrites, although those are going to be for the pseudo-unipolar ends, and we'll get to that. So this is going to be the most common neuron that you're going to find in the body, because this is the common neuron of motor neurons and the central nervous system. Bipolar neurons are going to be very rare. So they have the cell body in the middle, and then an axon going down one end with the axon terminals, and then they have the dendrite, a single dendrite pole coming off the other end with several branches for receptive fields at the other. Okay, so their two processes extend from the cell body. One is a fused dendrite, the other is an axon. So this allows for a longer dendrite, because in all likelihood the cell body is not going to fit here well for some reason, and we'll get to the reasons why. One of them is because this would be for special senses like vision and smell, and you go through these small channels that the cell body just is going to be too big for, or in vision it's going to get in the way. And then pseudo-unipolar, you have one process that extends from the cell body and it goes into the middle of the axon. Okay. Forms the central and peripheral processes, which together comprise an axon. So the axon is going from the dendrites over here, the receptive ends, all the way down to the axon terminal, and then you have a little tangent here that it goes up to the cell body. That's going to be in what's known as the dorsal root ganglia, the DRG. We're going to be talking about that when we get more into the spinal canal itself. Okay. So here are a couple of examples of where you might find these. So you can see the two different multipolar neurons here look completely different. One has that small axon that goes into the cell body, and then these dendrites will branch off in all different directions. Some are very, very long, some are going to be a little shorter, so that it can interact with as many neurons around it as it possibly can. And you actually, throughout your lifetime, will create new dendrites that can interact with other neurons that will allow you to learn and to retain information. So because you're not creating new neurons, you need to create new connections to be able to to change habits, to change function, to store memory. And then you have this Purkinje cell of the cerebellum, and you can see all of the branches that go up. And this would be known as like the arbor vitae, or the tree of life. You have all these neurons that are going to synapse with, again, more neurons within the cerebellum, which helps with balance and posture. The pyramidal cells are going to be found in the cerebral cortex. So this would be a neuron that would be for your conscious thought. The cerebellum is not conscious. It's going to be kind of the autopilot that we talked about last lecture. Because when you're standing, you don't have to think about all the different neurons that have to fire, all the different muscles that have to contract and relax. Your cerebellum kind of takes over and just does it. Of course, you have to learn that behavior as a child. Here are the two examples of bipolar. One, like we were saying, you have the olfactory cell. So here is the cell body, and then you have that dendrite that is going to lead to the receptive regions, and then the axon that is going to go all the way back into the brain. And then here you have the retinal cell. So the cell body is going to be in the blind spot of the eye. And then you have the dendrites that will go to the receptive regions, and then the axons that are going to go to the down the optic canal, and that's what allows you to create a picture in your brain. We're going to have a lot of fun discussing those two in the last two lectures of class. Special senses are going to be really the only place that you're going to see those bipolar neurons. So olfactory mucosa, which would be for smell. So olfaction is another word for smell. Eye for the retina, and then ear is going to be for hearing. So only special senses are going to be bipolar, and not all of them. Then the unipolar, or the pseudo-unipolar, is what it's more correctly called. So they're found mainly in the peripheral nervous system as sensory neurons. So this is not for your motor neurons, this is only for sensory. And the cell body is going to be found in what's known as a ganglia. Okay, so I had mentioned dorsal root ganglia, or the DRG. The dorsal root ganglia is going to be a collection of the cell bodies of all the pseudo-unipolar neurons at that level. So you have the receptive ending, which will be in your muscles or your skin, and it will come back through the peripheral neuron. It will have this little tangent just behind, or just posterior and lateral to the spinal cord, where you have this dorsal root ganglion. So all of these cell bodies are going to be in this little collection here, a little ball, and then the rest of the process, the axon, will go into the spinal cord and synapse, where it will relay its information to the spinal canal. Alright, so the functional classifications here. Sensory neurons are mostly in the peripheral nervous system, because you have to collect information from the periphery in order to bring that information to the brain for integration. So that would be your baroreceptors for pressure, your thermoreceptors for heat and cold, and it's also going to be receptors for pain, like a nociceptor. Those all have to collect information from the environment, from your body, to see what's happening, and they'll bring that information back from that area, like this muscle. Okay, it would come back into the spinal cord. So here's a better example from the skin. Send that impulse back through the peripheral nerve and into the spinal cord. And there is the pseudo-unipolar collection of cell bodies as the dorsal root ganglia. Okay, so it's a swelling here where you have all the cell bodies. And it's going to come in and synapse in the gray matter, which is where you have all of the cell bodies of the multipolar neurons, which would be your inner neurons that will integrate that information, possibly send it up the spinal cord for more collection or integration by the brain. So the motor neuron mostly is going to be in the central nervous system. So you have the cell body right here in the central nervous system, and it sends that information or the effect to the effector, which would be the muscle in this case. Okay? This here is the interneuron, or the association neuron. So that could be a neuron in the central nervous system that is relaying information from, say, the skin to lead to some sort of reflex. Or it could be collecting information from the brain and bringing it back to the motor to create some sort of response. So sensory neurons are really almost only in the peripheral nervous system. Okay, the motor neurons and interneurons are going to be almost completely in the central nervous system. So that's where the cell body of that neuron would be found. And then here you have the bipolar for the eye, for the ear, and for the nose. So the portion of an axon that communicates with its target cell is known as what? Okay, so the dendrite will collect information, the axon will take that information and send it down the stalk. The axon terminal is going to be the one that communicates with the target cell. And then the cell body is going to be what is where the nucleus is found, the endoplasmic reticulum, the mitochondria, etc., etc. So that's where all of the work is being done to create the proper environment for the cell. The nervous system has 3 overlapping functions. Which of the following represents a logical sequence of these 3 functions? Well, we did this with the last lecture, so what would make sense? If you are going to need to collect information, process that information, and then create some sort of response, you have to first sense that information, then you have to integrate it or figure out what to do with it, and then you lead to some sort of action or effect. Okay. So some of the terminology. Well, voltage is going to be one that we're going to discuss a lot because of the resting membrane potential. Voltage is going to be a measure of the potential energy inside and outside of the cell. So you will have a negative voltage within the cell because it has that large difference between the outside and inside. You have a lot of negative proteins within the neuron, within the cells, to create this environment. And potential energy means like taking a rock and putting it on the top of the hill. If you're sitting, if the rock is just sitting there, it has potential energy because as soon as you start to push it down the hill, gravity will take it and utilize that energy. So as soon as you open different voltage gates within the cell, you can actually create that energy shift and send that signal down the line. So the larger the gap, the more potential energy you have. Okay, so potential is voltage between 2 points. So the greater the potential, the greater the voltage. So negative 70 millivolts, which is the typical resting membrane potential of a cell, is going to allow for that energy flow. And now if you increase that to negative 90, now you have a larger potential, which means you have a larger voltage, which means that you can create a very strong signal. The current is the flow of that electrical charge. So think of that as a wave that is sent from the dendrites when it collects that information all the way down the axon to the axon terminal. So it sends that signal from one end of the neuron to the other end of the neuron and relates that information to something else. Okay. Another way to think about that, I explain to patients a lot, it's like a hose. So you have, when you turn on the hose, you have water that flows from the well or from the city water, and it flows down through the hose and then goes out the other end. Now, what I typically follow that up with is if there's interference in that, it's like kinking the hose, and it doesn't have that full signal going through. It really lessens the amount of water that gets through on the other end. So current would be the flow all the way through. Now, resistance is going to be a hindrance to the charge flow provided by substances through which the current must pass. Okay, so that would be, for the hose example, that would be like having mud stuck within the hose. That would be a resistance of some sort. It would decrease the amount of flow that can pass through the hose. And so again, you will get a lesser signal at the opposite end. Or you can think of it as kind of kinking that hose. So if you kink the hose, it doesn't allow all of the flow up to the end of the hose. So insulators will be high in resistance. Okay, so again, that would be like taking the hose and putting something inside of it, so that it has a smaller a smaller lumen, a smaller inside. There's less space within that hose for it to flow water properly. Conductors are going to be low resistance. They're actually going to help increase the flow, so that increase the current. That would be like standing in water, holding something electrical. Water is going to be a conductor. An insulator would be like a rubber, so rubber tires, etc. Okay. In our case, we're going to be using insulators like myelin. And I know what you're thinking, well, but I thought myelin is supposed to speed up that flow, and it does, because it allows you to skip portions of that neuron, so that now you don't have to go through the entire length, which could be several feet, but you skip inches at a time, which allows you to speed up that signal a lot. So Ohm's law is current, which is I equals the voltage divided by the resistance. Okay, so if you have a high potential energy and a low resistance, your current is going to be higher, which means your speed goes up. Okay, so the more resistance, the lower the current will be. Okay. As a general rule of thumb, the greater the potential difference in charge between 2 points, the blank the voltage. The higher the voltage. Okay, so the resting membrane potential, if you have it at -70, it's going to be a high voltage. But if you increase that to -90, which is would essentially be sending more positive ions out of the cell or bringing more negative ions in, the higher the voltage. So this is actually a common occurrence within neurons. You can actually take chloride and pump it into the neuron, which will increase the difference between the interstitial fluid and the neuron itself, which means that you have a larger gap or more potential energy. Or you can pump sodium into the cell, which will actually decrease the voltage, bring it closer to that threshold. So those are common uses where you're going to be able to bring that closer to or farther away from the threshold, which will be where you create that action potential. And speaking of action potential, this is where we're going to have to begin discussing what exactly happens to create a signal. So you're always going to get a movement down the electrochemical gradient. So there's always a movement from an area of high to low concentration, or in this case, opposite electrical charge. Okay, so a positive ion wants to go to a negative environment, and a negative ion wants to go to a positive environment. Opposites attract. It's just like a magnet. Okay, you can't. I'm sure you've done these experiments in school where you take magnets and you try to put positive end close to positive, and they repel each other. But you flip the magnet so that the negative end goes towards the positive, and they will increase the speed that they come together. Okay, so what we'll have are several different channels within the neuron. There's non-gated and gated channels. So non-gated would be like, okay, most people who live in an apartment complex don't have gates that allow you to go in and park in that complex. But in some of the very high-end apartments and condos, you're going to have gates that restrict who can come into that parking lot and who has to stay out. So essentially you have somebody that checks to see if you're supposed to be there, and if you're not, they keep you out. And that's what happens with these gated channels. They only open when the conditions are right. Non-gated channels will just always allow these ions to pass through. So they're also known as leakage channels. So they'll let some sodium leak into the neuron and some potassium to leak out of the neuron, but they will not allow this in high quantities. The gated channels are going to be either chemically gated, voltage gated, or mechanically gated. Okay, so what do all those mean? Well, chemically gated, also known as ligand gated, means that something has to bind to that gate to open it up. So these gates, like this one here, has a receptor region. Okay, so something has to bind to that receptor to open it up. When we discuss the neuromuscular junction, acetylcholine would bind to that receptor, open up those sodium channels, allowing sodium to get into the cell, and that would mean that you bring that muscle closer to threshold, which would allow you to create an action potential, which would allow for contraction of the muscle. In this case, it can open up these channels to allow sodium to get in, and again create that action potential if enough opened up for a long enough period of time. Voltage gated will be stimulated when you get to that threshold. So when you change the voltage, those channels will open. So this would be, for example, at negative 55 millivolts, which would be your threshold level. And so when you reach threshold, now you can open up those channels, okay, so that sodium can now rush into the cell and depolarize the cell, bringing it from negative 55 to a positive environment inside the cell. Now that voltage is going to be positive, which triggers a potassium channel to open. Okay, at that point, you will mechanically close this gate, which means that you actually put an arm in front of the gate so that it cannot open. So that now you completely block and do not allow that to open up. So mechanically would be a physical presence that closes off that gate. So chemicals and voltage will not alter that. Okay, so how do you generate this resting membrane potential? Well, you utilize these three ions: sodium, potassium, and chloride. Okay, the negative proteins that are sitting inside the cell are not going to move, they're not going to change. These three components can be easily changed to create the proper resting membrane potential. Okay, so you keep as much sodium outside of the cell as you can, as much potassium inside the cell, and then you also can keep chloride in. But you can alter these to make sure that you maintain the proper resting membrane potential. So you want to keep it somewhere between the negative 70 and negative 90 millivolts, so that it's close enough to reaching that threshold, but it's not so close that any little change is going to fire off an action potential. So you create this environment using a sodium-potassium pump. Now the sodium-potassium pump is going to pump sodium out of the cell and potassium into the cell. Okay, but why doesn't that keep things neutral? It's because you still have chloride, you still have those negative proteins inside the cell that keep a negative environment. Now you keep this sodium concentration high on the outside so that when you reach threshold, it rushes in. And then potassium will be inside, ready to rush out. Because if you have sodium as a positive ion outside the cell, and then you open a voltage-gated channel or a ligand-gated channel that allows it inside the cell, it has two really good reasons to get in. Because one, it goes from an area of high concentration to low concentration, and two, it goes from a positive environment outside of the cell to a negative environment inside the cell. Okay? Then, when you shift that from a negative 55 to a positive 30 millivolts inside the cell, now potassium, which is sitting in high concentration within the cell, really wants to get out because there's a high concentration inside, a low concentration outside, and a positive environment inside and a negative environment outside. Okay, so you're keeping that resting membrane potential and keeping that potential energy shift by using the sodium-potassium pump. So you pump sodium out and you pump potassium inside the cell to create this boundary. Okay. So here's a better explanation where we can get a little bit more in depth. So the sodium concentration higher outside the cell than inside the cell. So this is 140 millimoles of sodium outside of the cell and 15 inside the cell. Okay, and it's the opposite essentially for potassium, even though there's even less potassium outside the cell. So what you're going to do is you're going to push sodium outside and push potassium inside. And so you have to use ATP because you're pushing sodium and potassium against their gradients. So this is known as the sodium-potassium ATPase. Otherwise, it's taking ATP energy and utilizing it so that you can create that resting membrane potential. So it maintains the concentration of sodium and potassium across that membrane. Again, so you're keeping sodium out even though it really, really, really wants to get inside the cell. And you're keeping potassium in even though it really, really wants to get outside the cell. So you utilize that to create this environment. All right, so the permeabilities of sodium and potassium across the membrane are going to be a little bit different. So let's say that we just add potassium pumps, or not pumps, but leakage channels, so that a few potassium are allowed to sneak out. Okay, it's like opening a window in a dorm room. You get a couple people that can get out at a time. Okay, in this regard, potassium wants to get out, so a couple of them can sneak out and maintain this resting membrane potential. So if you allow some potassium to get out, it takes this negative 90 millivolts and allows that potassium to sneak out. Well, I should say it takes that negative 70 millivolts and allows it to go to negative 90. So because you're letting positive ions move from the negative environment to the positive environment, and that creates a more negative environment. Okay, you're essentially taking less positive, you're taking positives and sending them out into a more positive environment. So that increases the difference between the inside and outside of the cell. So potassium loss through abundant leakage channels establishes a negative membrane potential. So now we add in sodium leakage channels, which allow sodium to sneak into the inside of the cell. Okay, so that window's open, but now whoever is outside wants to get inside, and so the sodium wants to get into while the potassium wants to get out of the cell. So some sodium will sneak in, and that'll create a negative 70 millivolt, okay, which is now going to establish this push from sodium outside the cell to inside the cell, and some potassium from inside the cell to outside the cell. So now you have almost this even balance between sodium coming in and potassium going out. So that slightly reduces the negative membrane potential. So what we need to do is we add a sodium-potassium pump, or ATPase, to maintain the concentration gradients. So that keeps that resting membrane potential where it needs to be, and it can also help reestablish that negative 70 millivolt after you go through an action potential, which we're going to discuss in a few minutes. So the voltage displayed across the resting neuron membrane is termed the resting membrane potential. It allows for action potential and excitation. So the depolarized and excitatory potential is going to be kind of made up words, whereas the action potential is going to be when you actually get to the threshold, it sends that wave of the signal down the axon. So the signal is produced by the membrane potential. When you get this ion concentration and membrane permeability that can change the membrane potential, and it changes it at specific locations within the cell when you get these graded potentials. So graded potential will be any change in the voltage at a specific segment of that neuron, and typically that's going to be found at the dendrites. So what happens is you get this slight change, either negative or positive. It doesn't always have to bring it closer to threshold. A graded potential is going to slightly change the voltage one way or another. Okay, so you can have positive and you can have negative graded potentials. These can sum up to reach threshold or bring it farther away. Okay, so if you have a small graded potential that brings your concentration from negative 70 to negative 60, it still does not get to threshold, and if it doesn't get to threshold, you do not create an action potential. But let's say now you get this -70 to -55, well, now you're going to hit that action potential threshold, which allows that signal to pass through. So you need a buildup of those graded potentials to get to that point. But these graded potentials can also add more depolarization to the cell, which means that it brings it from that -70 to -80, -90, -100 millivolts, and that will make it more difficult to reach that threshold. Okay. So an action potential is only going to occur when you reach threshold. It's an all or nothing. So if you don't reach it, if you get to -56 millivolts, there's no action potential. If you get to -60, there's no action potential. You have to get to -55 millivolts in order to create this wave of propagation, okay, of that action potential. So if you get this point right here that has a stimulus that adds enough of a depolarization to get from -70 to -55, now you start this wave of change just at the cell membrane that brings the action potential positive voltage inside, and it propagates. So as you get to this point, well, now the original stimulus is going to be back to its normal depolarized state. Okay, so it's a very brief change in that voltage that allows for that wave, sending a signal. Okay. So depolarization, break down that word. Okay, polarization is going to be establishing opposite ends. So the north and south pole of the Earth are going to have be the two extreme ends of the planet. Okay, so when we depolarize it, we are taking those poles and bringing them closer together. Okay, it's like as if we could kind of squish that ball and take the north and south pole and bring them closer together. Okay, we're establishing an equilibrium within that environment. Hyperpolarization is going to take the resting membrane potential and bring it farther away from threshold. Okay, so it's almost like taking that same ball and squishing it so that the north and south poles are going to be spread farther apart. So in this case, you're going to make the inside of the cell more negative. In depolarization, you make the inside of the cell less negative or more positive. Okay, so the graded membrane potentials are going to be short-lived, localized changes in the membrane potential. That magnitude will vary with the stimulus strength. Okay, so the stronger the stimulus, the more likely you get to that threshold, the more likely that you get to the action potential creation. Okay, so if you have a very weak signal or very weak stimulus, chances are that you're going to get a little change in the environment in that one area, and then nothing happens because you haven't reached that threshold. Now you increase the stimulus more and more and more until you get to threshold. Then you start to propagate that signal by the action potential. Okay, so there could be a stimulus that opens up a sodium channel, but it only takes that from -70 to -60. Well, you're not going to create an action potential because you haven't reached threshold. Okay, now let's say the resting membrane potential here is at -90, and you change or depolarize it by 20 millivolts. Do you reach a action potential? No, because you only get it back to -70 millivolts. So it doesn't reach threshold, which is at -55. Okay, if you take -90 and then you depolarize it by 30 millivolts, you get to action potential. No, again, you only get to -60. So if you're at your resting membrane potential of -70 and you depolarize by 30, well, yes, you've gotten to threshold at -55 and you create that action potential. So it's an all or nothing. You either get to threshold and create that action potential, or you don't get there and action potential does not occur. So these are the different components of the action potential. So we're going to talk briefly about this, and then we're going to take a little break and create a new video to discuss the rest of the lecture. So the resting state is going to be where the cell is doing its normal activities. So no action potential is occurring. You're maintaining that resting membrane potential, give or take a few. Um, and you need some sort of stimulus which will create that depolarization to bring it to threshold. So it's doing its job, doing its job, doing its job. Now a neuron is interacting and it's opening up those ligand-gated sodium channels. It comes up, gets the threshold, boom. Now you go through this action potential, which involves depolarization. So you're bringing it to closer to zero, closer to that equilibrium. It overshoots that and goes to positive 30. And now at this point, well, you want to bring it back to its resting membrane potential. So you close the sodium channels and you open the potassium channels. Yes, this is a lot of information, but I'm going to briefly go through this and then we'll talk a little bit more about that in depth. Now you go through this repolarization phase where you now are going to separate those poles. Now you're going to have the negative balance inside the cell and the positive outside of the cell. So you're bringing sodium back from the inside to the outside of the cell, creating that negative internal environment. So repolarization is bringing it back to its resting membrane potential, and it overshoots it again where your resting membrane potential is, which is known as hyperpolarization. So it brings it past the resting membrane potential, and now you go through that repolarization or hyperpolarization phase to balance it out to its resting membrane potential. So all this will happen within milliseconds. You can see the time here, milliseconds, less than 2 to really create that action potential. And then you repolarize and reestablish that membrane potential. So if you don't get to threshold, if you get that signal right here, you're not going through that action potential, and it comes right back down here. There's no signal sent. There's no propagation of that wave. So you either get to threshold or you don't. You either create that action potential by getting to threshold or you don't. Okay, so we're going to discuss this in the next set of lectures in depth. I want you to